funciona. Sí funciona el micrófono.
Blank, ¿verdad? Sí? Okay. Well, good afternoon. Thank you everyone to be here in our first official seminar in the South Campus of Simvesta. So for me it's a pleasure, a real pleasure, to present you Dr. Uli Blank. Dr. Blank comes from the Laboratoire d'Excellence Inflamex, of the Center for Research in Inflammation in the INSERM U1149 in Paris, France. Dr. Blank obtained his first grade in biology and human biochemistry in Germany. And after that, he moved to Paris to study mast cells. In the first studies he did in his master thesis, he studied the production of IgE in humans. And he identified different patterns of production of IgE in humans. Then he moved to study the cell that was responsible for allergic reactions, that means mast cells. And during his very long and productive career, he has been every few years making important contributions to the field, to the study of allergies, mast cell physiology, and the structure of the IgE receptor. Just one example, he was the first one into transfecting the mouse IgE receptor in COS7 cells, and proved for the first time that that receptor was able to bind IgE, and that antigen was able to cross-link the receptor and induce the secretion of pro-inflammatory mediators. After that, he moved to study a little bit of gene regulation of cytokine production. And finally, in the last, I think, more than 20 years, he has been studying the process of secretion in mast cells. And he has identified new proteins and the mechanism by which these cells can perform mediators and to induce inflammation. As you know, inflammation is behind many chronic diseases and in inflammation too. So as students and researchers, we have to learn about inflammation, and I really want to thank Uli to be here to show you something. Thank you, Uli.
Okay. Thank you, Claudia, for these nice words, and I'm really happy to be here third time in Mexico, but I'm very well acquainted with Mexico because in France we had many students and postdocs from Mexico, so they have been accompanying my career until the very end, because since I'm June last year I'm status, so I'm officially retired, but I'm still in science and try to continue as much as I can. Okay, so today I will give you a sort of an overview of what we have done in mast cell signaling, looking at mediator secretion, and it's an update of about 20 years of research in that field. Now, mast cells are very important cells. They can release a variety of mediators when they are stimulated, for example, via the IgE receptor. And these mediators are implicated in tissue responses, vasodilatation, bronchoconstriction, mucus secretion, for example, with histamine. They maintain the acute and chronic inflammatory response by attracting other inflammatory cells. They can also regulate immune cells via release cytokines and participate in tissue and repair remodeling. I've done a mistake. It comes back. I shouldn't put.
remodeling responses via specific proteases, growth factors they release. Now, mast cells are pretty much known for that because mast cells are bad cells. They cause allergies, and about 30% of the population has allergies. The most severe form is anaphylaxis, less severe rhinitis, asthma. But the good side, and this came out only in the last 15, 20 years, they have also good sides. They are very important in the defense against environmental toxins. For example, snake venoms or are pieces of xenobiotics, irritants. They can actually, via the action of proteases, inactivate or via, for example, via heparin release, neutralize these substances. They are also anti-infectious cells because they can fight parasites, bacteria, fungi, viruses via express toll-like receptors, for example, but also via adaptive immunity. Sometimes they can overreact and cause detrimental function. They are also contributing to chronic inflammatory diseases and autoimmune diseases. That's again a bad side, but they, as I already mentioned, can also participate in tissue repair and immunoregulatory responses. Now, most of the people, most of the immunologists, they ignore mast cells, and I just have put this last night, this slide from our RMB mouse, red mast cell and basophil mouse, to show you that they are really very frequent cells in
tissue. When you count them, they are up to 300 to 400 cells per square millimeter in the skin, almost as much as Langerhans cells, which are much better known in the immunological community. Also, mast cells now, at least some immunologists appreciate them.
But we have been interested most of the time in studying signaling. Many people via the IgE receptor. This receptor is composed of an alpha chain, beta chain, and two desulfide link gamma chains. And when you cross-link this receptor or aggregate with a multivalent antigen, you trigger responses such as mast cell degranulation, cytokine, chemokine production, and lipid mediator production, prostaglandins, leukodrieins. Many people looked at the early signaling events, but coming back to France from the US, I don't want to do what other people do, so I said I go rather looking at the late events
governing the degranulation response. And this is how it looks like. So this is
a mast cell. You see it's full of these secretory granules with this proteoglycan electrodense matrix. And when you stimulate them, we do this with a monoclonal IgE directed to the DNP hub 10, and then we come with the DNP human serum albumin to cross-link the receptor. And what you see is really a very dramatic response. The mast cells essentially can release up to 100% of this granular content, and you see these pre-stoned mediators are released, histamine, tryptase, but they can also contribute to synthesis of lipid mediators and chemokines and cytokines. And when you look closer in this electron microscopic picture, you see that there are a lot of membrane fusion events going on. So we became interested in studying these membrane fusion events, just to say we measure these degranulation response either by histamine release, measuring beta-hex release, or also CD63 externalization, which can be evaluated by FACS. Now, fusion processes have been, is mediated by proteins which have been discovered in the neuronal system and have been to snare proteins. You have the vesicular snares like the VAMP family of proteins and two T-snare target snare families, the SNAP2325 family and the syntactine family. And what they do is actually they can form a very stable tetrameric trans-snare complex. The formation of this complex is energetically favored, and this actually drives membrane fusion. Now, the energy requiring step in the whole process is actually the dissociation of this trans-snare complex. You have the binding of an ATPase via an adapter protein, which binds to this complex and can reconstitute the individual snare proteins. And this is how it looks like this is the crystal structure of the neuronal snare complex. You have 4 highly intertwined helices of about 60 amino acids, and they are actually the substrate of these famous and bacterial toxins like Botox.
And they can be dangerous, but they are also employed by famous actors and artists.
They don't look as nice as before. Concerning mast cells, the first one was really David Castle to show that these snare proteins are implicated in the degranulation process. He looked at SNAP23. You see it's on the plasma membrane, and when you stimulate the cells, they go into the interior, so there is a relocation of SNAP23. And when he introduced a blocking antibody in permeabilized peritoneal mast cells, he could block this degranulation process. On our side, we studied the syntaxine family members. You see, for syntaxine 4, we have more or less the same pattern on the plasma membrane, and when we stimulate, there's an inward movement. But on the other hand, the syntaxine 3 isoform is on the granules, and there's an outward movement to the plasma membrane when stimulated. So we tested the implication using an anti-knockdown approach. You see here the degree of down regulation, and when we down regulate either syntaxine 3 or syntaxine 4, we get a significant inhibition of the degranulation response. Concerning the V-snare, we concentrated on this VAMP8 isoform, and the reason for is that VAMP8 is also called endobrevin, was initially discovered to be localized on endosomes, and when you granules from hematopoietic cells are actually not specialized secretory granules, but they are so-called lysosomal origin. So there is a connection with the endosomal lysosomal system. So that's why we concentrated on the VAMP8. And the other reason was there was a knockout strain available, and we collaborated with Wan Chin Hong in Singapore, and as you see here, in these knockout strain, they are degranulation deficient, they don't release as much histamine, and when we test this in an in vivo passive systemic anaphylaxis model, you see there is much less histamine released in these mice. Another important point was we were really the first to show that there is really snare complex formation after stimulation. Already at 1 minute you can co-immunoprecipitate the WAMP8 protein with an anti-SNAP23 or with an anti-syntaxin 4 antibody. So clearly we have snare complex formation in these cells. Now, I told you that this complex formation is energetically favored, and it can actually happen without any regulation, but in living cells usually it is regulated, and you have positive and negative regulators. I will give you two examples. The first example is Iris' example, because when she was in France, she studied this thomosine molecule or syntaxin binding protein 5. It's like the WAMP protein. It can also bind to the T-snare proteins, but as you will see, it is negative regulated. And the reason why we looked into this is that thomosine was really very highly expressed in mast cells as compared to other cells by these bio-GPS data. When you look now in the INSERM database, there is also, for example, eosinophils, which very highly expressed this thomosine molecule. So thomosine is a very large molecule, 110 kilodalton, and the C-terminus, it has this R-snare or V-snare domain, and then the N-terminus, you have these repeated tryptophan aspartic acid domains, 7 of which, or 2 times, and they form this beta propeller structure, and this is actually a protein interaction platform and also serves to oligomerize the thomosine. And the data which have been published were in the neuronal system in the knockout mice. You have actually enhanced synaptic transmission. But Tanaka never sent us the mice after repeated contact. We gave up, and we used, rather than this knockdown approach we were already used to. So you see the degree of down regulation, and interestingly we find the same data as in the neuronal system, that in the absence of this thomosine protein you have enhanced degranulation, here measured by CD63 exposure or by beta-hex aminodase release. So it really serves as a fusion clamp regulator. And then we got a little bit more into the potential mechanism, and as you can see here nicely, when we immunoprecipitate the thomosine with co-immunoprecipitate with an anti-syntaxin 4 antibody, you see that after stimulation you see a decrease in association, so it dissociates from the syntaxin 4 molecule. But to our surprise, the contrary was the case with the syntaxin 3. But as you see, this is a little bit delayed as compared to the dissociation. So maybe there may be some different function associated with it. And Iris also found that it gets phosphorylated, not on tyrosine, but she used a phosphothreonine antibody, and you see after stimulation you get a nice phosphothreonine phosphorylation, and then we looked at what kinase could be responsible for that using inhibitors, a PK protein kinase inhibitor, because it was already shown that it can be phosphorylated by a pKa, but this did not have any effect. On the other hand, protein kinase C inhibitor had a strong inhibitory effect on this phosphorylation. So we looked what isoform of PKC isoform was responsible for that, and we focused on PKC delta because this phosphorylation was calcium independent, and PKC delta is a calcium independent PKC isoform, and we did again the knockdown approach, and you see the PKC delta can decrease, downregulation can decrease phosphorylation. But interestingly, also as a consequence of the inhibition of PKC delta activity, we don't see again this dissociation of thymosine from the syntaxin 4 molecule, but also the association with the syntaxin 3 was blocked in the system. So this is our model of regulation. We have the thymosine molecule bound to this syntaxin 4 SNAP23 complex. When you stimulate the cells via IgE, it will dissociate, and this will allow the fusion process. So when it's not phosphorylated, it acts as a fusion clamp. And what we think happens after that, it's maybe the phosphorylated form then may be able to bind to the syntaxin 3 complex, which is on the secretory granules, and this may sort of be a secondary fusion clamp. It say to the mast cell, hey, stop, don't go on, stop your degranulation, because this can be dangerous to the body. Concerning the positive regulator, I want to tell you the story about this transport molecule, the kinesin 1, which is a motor protein involved in microtubule transport. And actually the coordination with the cytoskeleton is actually a very important aspect in the degranulation process. And I always show this very old, already in the 1980s, produced electron microscopic picture. You see that granules are not alone. They are really embedded in a meshwork of cytoskeletal elements. So clearly there has to be coordination between the IgE stimulation and remodeling of this cytokine, not cytokine, actin and microfilament elements. Also, there is transport processes happening. You see the granules labeled here with CD63. When you stimulate the cells, they go to the plasma membrane, and when you block microtubular remodeling with nocardazole or with Taxol, you abolish this membrane movement of the CD63 molecule. So one of the transport important motor proteins are kinesin. There are many different members, but we concentrated on this kinesin molecule because we found that it is expressed, the KIF5B isoform is expressed in mast cells together with the kinesin light chain. It's actually a tetramer, 2 heavy chains and 2 light chains. At the end terminus you have this motor domain which binds the ATP and drives forward the movement. And now Gail Menashe, 2 blocks from our lab, she had available these floxed K5B mice, and we crossed them with hematopoietic specific WAF-Cray mice, and when we grew bone marrow derived mast cells, primary mast cells, you see they do not express the kinesin 1 molecule, and at the same time they are deficient in degranulation, here measured by beta hex, and also passive systemic anaphylaxis, here measured by the drop in rectal temperature when you challenge IgE sensitized mice with antigen. And you also nicely see when we label the granules with a fluorescent marker, WVGA, you see we do this by TIRF, total internal reflection microscopy, where you can look into the first 100 nanometers of the cell. And when you stimulate them, you see the granules pop up at already about 8 minutes, and they continue to do so. In the kinesin knockout, you see there is essentially no popping up. It comes out a little bit at the end. So the granule movement is restricted in these mice. Now, how do these molecules connect? Well, we knew already that there is a small GTPase RAP27 which had a role in granule transport. This was published before by Miguel Seabra's group, and then we looked at RAP27 effectors, and these are the synaptotacmin-like proteins which have this SHD domain, which is nothing else than a RAP27 binding domain. So we just had to look what kind of isoform is expressed in mast cell. And as you see here, we have a nice expression of the SLP3 isoform. So we tested this, go get a little bit more into detail. So we did labeling studies. You see RAP27 is on the granules, but also this SLP3 on the granules together with the granule marker. They are colocalized very nicely, and they go in concert to the plasma membrane once the cells are stimulated. So again, we use this knockdown approach of SLP23, and so when we knock down, you see that there is a down regulation of the degranulation response. So we can see that the kinesin 1 molecule gets connected to the RAP27 via this SLP3 connecting molecules. We can also co-immunoprecipitate this complex. I don't have the time to go into this, but this is the summary of all these molecules which regulate the fusion process. If you want to read more, we have written a review. Now, in the last part of my talk, I want to go a little bit more into, not into the degranulation process, but mast cells produce also neosynthesized cytokines and chemokines. And our strategy was again based, looking which one we shall go for. I don't know whether you can see that nicely. We choose the MCP1 monocyte chemoattractant protein, now called CCL2 chemokine, because it's very highly expressed in mast cells and not so much in other cells. And CCL2 is an important chemokine attracting essentially monocytes, which then fulfill different biological function, but it's also an important chemoattractant, for example, for for basophils. Basophils during an inflammatory response very often infiltrate the tissues. Eosinophils express the receptor, but also neutrophils are responsive to CCL2. Now, little is still known for chemokine or cytokine trafficking. The first one really were doing this were Jennifer Stowe's group in Australia, and she found actually in monocyte macrophages that, for example, IL-10 gets directly to the plasma membrane from the Golgi once synthesized, whereas others like TNF or IL-6 intersect in the so-called recycling endosome, and from there it gets sorted from there to the plasma membrane. For TNF, IL-6, IL-10 gets also, so they have different pathways, but for example, TNF can be specifically sorted to the phagosome in these macrophages. In dendritic cells, it was published that IL-12, a very important cytokine for driving Th1 differentiation, intersects VAMP7 vesicular, so lysosomal compartment here in multidirectional, and it was very strongly dependent in this polarized secretion when it forms synapse with the T cells. Concerning T cells, Mark Davies' group has shown that some cytokines go directly to the synapse, like IL-2, interferon, gamma, IL-10, whereas other multidirectional get secreted all over the place. So what about mast cells? So the first thing we did an immunofluorescence study, and we could see that already in non-stimulated cells there is some CCL2 stored. These are the peritoneal derived primary mast cells, and this is the bone marrow derived mast cells. So the PDMCs, they contain a little bit more. And then new synthesis comes up. You see then the fluorescence increases, and at 3 hours it decreases, which is in agreement with its potential secretion. And when we measure this by ELISA, we actually see 2 waves of secretion, a very early one mediated probably by the degranulation process, and then new synthesis comes up, and CCL2 gets secreted via after having been newly synthesized. And we confirm this pattern by using this transcription inhibitor actinomycin, whereas actinomycin completely blocks the secretion of IL-6, meaning that it's only secreted as a newly synthesized protein. It only partially blocks CCL2 secretion, meaning that some of it gets released by degranulation. And we actually looked at the co-localization in these non-stimulated cells, and you see there is a nice co-localization with the MCPT6 genuine granular marker in these cells. So really there is some CCL2 stored in these granules. Now what about the newly synthesized one? How does it traffic to the plasma membrane? Is it using an IL-10 pathway going directly? Does it intersect with a recycling endosome like TNF? Or does it go via lysosomal compartment via the IL-12 in dendritic cells? So we explored that by looking at co-localization, the VAMP3 recycling endosome pathway. There was very little co-localization. Also some cells, not all of them had a nice co-localization. But we saw much more co-localization, especially after stimulation with a VAMP7 compartment up to 40% co-localization, meaning that there is maybe this IL-12 pathway like in dendritic cells. And we tested this using VAMP7-KO mice together with, in a collaboration with Terry Galli's lab, who gave us the VAMP7-KO cells. And we could actually see that CCL2 secretion is inhibited in these cells. Now we wanted to see a little bit more, also dynamically, how this CCL2 behaves. So we made a new mouse strain, a reporter mouse strain, where we coupled this EGFP to the C-terminus at the stop codon to to produce a CCL2 EGFP fusion proteins. The mast cells grown from these mice develop normally. They express the IgE receptor and the c-KIT marker. They degranulate correctly, like the wild type cells. So these mast cells seem to be functional. They also produce CCL2. They have pre-stored and enhanced production after stimulation, and after 3 hours they lose the expression due to secretion. And we also have this two-wave pattern of secretion. Now Eduardo, the next Mexican student, he then designed this live imaging assay where he put the cells into medium which contain this avidine sulforhodamine, which actually can bind to the proteoglycans released during the degranulation process, and we can therefore measure also monitor the degranulation, and the EGFP will be fluorescent, and you see that about 40 to 50% of the non-stimulated cells contain these pre-stored EGFP. Some of them contain really pretty much, and this is even more in the PDMCs, which have a nicer granular compartment as the BMMC. Now, when we launch the movie, you see that in the beginning just the red is degranulation, but then comes up the EGFP synthesis, and you see actually different ways. So just in the beginning red, then you see the Golgi passage, and then you see the EGFP goes out either multidirectionally or in some cells. This, for example, when you look at this one, it's very striking. It has sort of a polarized secretion. It goes into one direction. Another observation we made in these movies is once the chemokine goes out, it very nicely co-localizes with the released proteoglycan matrix, and this is known. All chemokines more or less have glycosaminoglycan binding ability, and heparin, which is a major constituent of the PDMCs, is very nicely binding to this CCL2. But now Eduardo, he tried to quantify the patterns of release from the individual cells. So we designed the multidirectional pattern, polarized pattern, and very interesting pattern which we call the pseudopodia-like pattern when you have here a mast cell which really adheres very strongly to the surface. We saw this pattern that the chemokine really goes into the pseudopodia like structures. But most of the cytokine is more or less released multi-directionally, but some of it also gets secreted in a polarized manner. Now, this made us again think about this must be regulated somehow by the cytoskeleton and movement. And so we again looked at microtubular tracks and actin cytoskeleton. The actin cytoskeleton makes this actin ring in non-stimulated cells and is maybe responsible for perpendicular movement via these myosin motor proteins. You know already about the kinesin, which moves to the plus end, but there is also dynein, which directed to the minus end. And we used drugs, nocodazole and cytochalasin D, which affect either the microtubules or the actin filaments. And when we do this, you see the actin ring in control treated cells is completely dissolved in the cytochalasin treated cells, and the tubulin pattern, when you stimulate the cells, they stretch out, and when you add the nocodazole, it dissolves, and these tubular tracks cannot form. Now, cytochalasin D does not really have an impact on CCL2 secretion, whereas there's a small effect of the nocodazole, and we think this is due to the because it strongly inhibits degranulation, and so this inhibition may come from the effect on the pre-stored CCL2 release. But these drugs had very important effects on the secretion pattern. When we inhibit microtubules with nocodazole, we saw much more cells forming these polarized secretion pattern. When we inhibit the actin cytoskeleton, or no, I'm sorry, this is multidirectional when we knock down, and when we knock down actin, the cells make much more polarized because they only have the tubular tracks where they can move on. So the pseudopodia-like pattern is completely abolished because the cells cannot do this pseudopodia anymore. Another important feature of chemokine or mast cells is that they are also able to polarized directional secretion, and this has been already shown in the 70s when mast cell is stimulated with beads which have concavalin, which aggregates the receptor. You see they degranulate into one direction, and Eric Espinosa has taken this up in a very nice study where he looked at Toxoplasma gondii parasite and human mast cells, and when the Toxoplasma gondii tries to infect the mast cell, and the mast cell is given anti-toxoplasma antibodies, you see you have a very nice local degranulation response, probably to increase the efficiency of anti-parasitic action. And he could actually show that the proteases like kinase can participate in killing the parasite. Now, mast cells are involved in anti-parasitic action, maybe via these degranulatory synapse, as Eric Espinosa has called it. But how do chemokines and cytokines behave? So Eduardo again designed up a new assay of a polarized degranulation system where he coated the antigen on the LabTech surface. It took him a half a year to put to get this going, because when you coat the antigen, it actually leaks, and you don't really see a nice degranulatory synapse. And he had to use a product called CellTAC, which is actually from mussels. It's the product where mussels get fixed to the stones, and when you do that, the antigen really sticks to the surface and does not leak, and you can actually really see a very nice degranulatory synapse. The procedure is a little bit different. You have the antigen coated, so you add the cells, but they have to go to the bottom, so it takes about 20-25 minutes to go down to reach the antigen, and then they start to form this very nice degranulatory synapse. But as you can also already appreciate, the GFP, the chemokine, rather goes or has a tendency to go into the opposite direction. And now he tried to quantify that. He divided the cell into 4 regions. Region A is really the one at the degranulatory LabTech surface, then region B, region C, and region D, which is the most opposite one of the degranulatory surface. And then he did for every cell this fluorescence pattern. You see in non-stimulated cell the GFP CCL2 found. There is a tendency at the early time points that it goes to the degranulatory synapse, whereas at later time points there is the tendency to rather go to the opposite direction, and he did this for many, I think 30 cells or something like this. You see this nice drop in the early time, so some of the GFP fluorescence goes down, and then the newly synthesized has the tendency really to go up to region C and D. We didn't really quantify the B region because this is the region where the Golgi is located, and it's found there. Now we also started to go a little bit in vivo. We did this PCA experiment. You sensitize the mice with IgE in the left ear, control is PBS in the right ear, and then you challenge intravenously with the antigen, and then you analyze the fluorescence, essentially. Unfortunately, we couldn't use GFP fluorescence because when you fix the cells, and we are not yet set up really for in vivo imaging, so we had to cut the ear and fix it, so you lose GFP fluorescence, but we can very nicely detect the CCL2 is here in white, the CD31, which is the vascular, the blood vessels, and IgE in green. And you see the isotype antibodies don't give a signal, but we had sort of a problem is when you inject PBS in the ear or IgE in the ear, it already creates an inflammation. So even 24 hours after having injected PBS, we still saw some basal CCL2 in the area, which was sort of a problem. But when we then challenged the mice, we nevertheless saw a significant increase. And Eduardo, he did two things. He looked at blood vessels, so co-localization with the CD31 marker, and the CCL2 in tissue. And you may be, it's not big enough, but when you look close, you have here a blood vessel, and the CCL2 really aligns on this blood vessel, so it sticks to the blood vessel, whereas after longer time points we also see it in the tissue, and at 120 minutes is more or less gone. You see, there's less of a problem with the PBS in the tissue, but with the IgE we still have some background. But the point is that this CCL2, once released, and mast cell actually can go through these endothelial cell layer and actually degranulate into the bloodstream. This has already been shown by Andudek for TNF. But the CCL2, because it has this proteoglycan binding capacity, may be immediately captured by these glycosaminoglycans expressed by these endothelial cells, because it has proteoglycan binding affinity. This may induce some signaling which may favor adhesion molecule expression, and this may be important for local or attracting the inflammatory cells to the local inflammatory site. And this is the thesis summary of Eduardo. So we have the IgE receptor stimulated mast cell. They degranulate into the bloodstream. The CCL2 gets captured by the local syndecan proteoglycan molecules, and these foster adhesion molecule expression, and then neutrophils, monocytes, eosinophils may come up and get attached to this vasculature, and from there on they go into the tissue to fight infection or whatever, to maintain the inflammatory response, and maybe other cells than mast cells may take over to maintain the inflammation. That's it. Maybe I've been a little bit too long. These are all the people who during the years have participated. Eduardo, he has now switched to the Imagine Institute after the thesis. We still have to publish his work. Iris, who is here, a Mexican student. There is, I think, Chanja is here. She was there for 6 months. Carla is not here. And so we had a couple of Mexican students visiting, and of course the project with the CCL2 couldn't have been done with Claudia, because we got a grant together to make these CCL2 EGFP mice, and then of course long-term collaborate John Rivera at the NIH, Joanna with the Tomosine, and Gael and Isabel Munoz with for the kinesin study. And that's it.
I have a question regarding the cytokines and the loading of the cytokines into vesicles. Is this specific? So are there vesicles for each one, or of the cytokines?
For each cytokine?
Yeah, or maybe they are secreted together, some of them, I don't know. But is this a specific mechanism to loading the vesicles?
I think it's not yet completely known. As I have shown, for example, IL-10 in macrophages, it can go directly to the plasma membrane without intersecting any specific vesicle. I think it just pops up from the Golgi apparatus and goes there. Others, like TNF or IL-6, they go to the RAM3 recycling endosome. And in our case, I think there is from the Golgi it goes to lysosomal compartments, but whether there are still other intermediates, we don't know. And the other thing, for example, in T cells, Mark Davies, he has described IL-2 interferon gamma goes to the synapse with the dendritic cell, but TNF goes. What governs this pattern?
We don't know.
I don't really know. Even he does not know, I think.
And so this kinesin 1 molecule, this system, is specific for mast cells, or it happens in any secretory?
For the CCL2. There are some other cells, for example, monocytes themselves can produce CCL2, but we haven't looked in monocytes, because we could have looked in other cells, but not yet done.
Okay, thank you.
Other questions?
Hi, thank you for the talk. I was wondering if the process of mast cell activation can be influenced or changed in any way under hypoxia, for example, in organisms residing at high altitude, or during an infection where respiratory function might be compromised?
Mast cells can be activated by many different, they can be activated by parasites, they can be activated by a G protein coupled receptors, which has been very much studied here in the institute, and by by cytokines. IL-33 is a very important co-stimulator, and I think all these receptors can work together to make the mast cell more efficient. But on the other hand, as you know, it can also be dangerous, because if you release too many mediators, you can have allergies and even anaphylaxis, which can be deadly. So, for example, I haven't shown this for the thomosine molecule. Iris, she did some studies with patients, and what she actually found is that the thomosine molecule gets expressed in people who have very high IgE levels, so very highly allergic people, they express more of this tomosine, and we think that this may be acting as a degranulation brake, which gets upregulated in highly allergic people. But the inflammatory response in general by mast cells, I mean, in an in vivo setting, there's not only IgE, there is maybe many other products which still can co-activate the cells.
Oh, thank you.
I just want to ask you, sorry.
In the very
Very nice talk. I'd like to, well, I have several questions. It's a very good chance for me to learn about mast cells. But I would like to start asking about the CXCL2 chemokine. How is transported from the tissue to the lumen of the endothelial cells? Transported, because the endothelial cells don't allow the migration is exactly.
I start with the vasculature. I mean, really in the mast cell, they can really reach through the endothelial cells and degranulate into the vasculature. And for example, the Andudek has shown for the TNF, it gets actually transported into the bloodstream away and gets disseminated. For the CCL2, we think, as we showed, it stays there because it's captured by the syndicans. Now, in the within the tissue, I think it does not travel. It's just in the surrounding, and maybe it can also bind to the heparin, which is released by degranulation. But I don't think it travels a lot. It just stays in the area.
Yeah, in fact, I was thinking. So the mast cells is opening the endothelial cells, so it requires molecules in order to open those tight junctions between the endothelial cells in order to deliver the chemokine in the bloodstream. That's right.
Okay. The blood vessels. I mean, they release histamine. I mean, histamine is one of the most important substances which can open the blood because the endothelial cells they express the H1 receptor, but there are also I don't remember if it's prostaglandins or leukotrienes. There is they also release platelet activating. All of these can open up the blood vessels. But the most important one is histamine. When you have an anaphylactic shock, it's mediated by histamine.
Not related with your talk, but I know you may know the answer. Is there any populations of mast cells? I mean, you have mast cells in all the mucosas, and not all of them behave exactly the same. For example, we have the ones that are regulating allergic responses, for example, in the lung, but the ones that are in the intestine, for example, are more related to as a mechanism of defense to parasitic diseases. So is like, for example, in macrophages, in other cells, that you have a population more inflammatory and another population that is, I wouldn't like the word anti-inflammatory, but more regulatory, with more regulatory functions. Can you see difference on those cells, or they do exist?
It's about the heterogeneity. I'm not really working on that, but I mean, in the 70s, it was much simpler than now. 50. In the 70s, they had this dichotomy of mucosal type mast cells and connective tissue type mast cells, which were, I mean, Ennebeck, they had different staining properties. He described that. But when you look now, when the publications come up with the single cell RNA sequencing, now they make mast cell population 1 to mast cell population 6. All of them have different kinds of mediators. But the problem, I mean, it won't be my business anymore to find out what does mast cell population 2 or 5 in real biology. And I mean, this is also something coming up for many other macrophage population. They find so many subpopulations. Is this really biologically relevant? I don't know. Well,
thank you. Thank you very much.
Any other questions? I will ask you about therapy. Because when we are studying mast cells, we are all the time saying that this sometimes is going to be useful for treating allergies. And for example, for the students, how are the new strategies to target real mast cell activation and allergy? You have to remove the IgE, for example, that's one strategy. But one of these molecules that you have studied can be considered as a marker or as a therapeutic target?
I mean, the anti-IgE strategy, in my opinion, was the good one right away. I mean, I agree, all these molecules could, I mean, one initial idea we had is, for example, the VAMP8 molecule, which affects degranulation and maybe not cytokine chemokine production, so that we have selective inhibition of the degranulation process as compared to chemokine cytokine secretion, which may be useful because the mast cell can attract other cells to the inflammatory site and just block the allergic response. But the problem with all these snare molecules is that they are pretty ubiquitous. The only one maybe is the tomosine. You saw it's highly expressed in mast cells, but the newer data say also eosinophils, but still maybe foster sort of a fusion clamp binding to syntaxin 4 and blocking fusion is still an option, but it has to be a big protein, at least 60 amino acid to bind to and block. So it's, or maybe enhance like high IgE allergic people do, the tomosine expression. But more or less all these NER molecules are, and also the regulatory molecules are pretty ubiquitous. So I think the IgE strategy works very, very well.
Thank you very much. Thank you.